Well, my week started with invigilation. I was among the PG students that were assigned to invigilate the final year students. They wrote their final exams on Monday, and it was a successful invigilation. Immediately after the invigilation, we went to Animal House in Health Sciences to get a rabbit for our experiment. Unfortunately, we couldn't get the rabbit that we needed there, so we had to go to another location outside the school to look for rabbit. And then we saw this different genders of rabbits, and we decided to take the male. This was the rabbit in the cage. I had to transport it from that location to the school. Now that was the summary of what we did that day. The next morning we were informed that we'll be dedicating the molecular biology lab in our department. Remember, I told you that the department is biochemistry and molecular biology. So before today, there was no molecular biology lab, just biochemistry lab. So a particular individual, funded the, restructuring of this lab, introduced important equipment for molecular biology practicals. And it was good because this particular day, this is the VC speaking. The VC came and dedicated the molecular biology lab. This is the name of the individual in that particular place that funded the project. Now this is the lab. Even though it's still under construction, they are still imputing important equipment. In fact, we're even having lectures today, and the lecturer was telling us about the lab, how it's going to help us, how it's going to make the research amazing and all. So we went around the lab, and then it was very, very vital that we see some of these modern equipment, important equipment in the lab. Hopefully, as we progress, I will show you the finished work. But for now, this is where it is, and these are PG students. These individuals are seen here, they are all PG students. We came in to see the lab and all that. Now, one important thing that stood out for me is not just the dedication of the lab, but how everyone came together for this humble occasion, from the HOD to the different professors and lecturers in the department. And then we look at the administration, like the VC, the DVC, the chief security officer. Every stakeholder in the institution came around for this particular dedication, and it was amazing. Do you understand? It was very, very amazing, to the point that we even had an after occasion. This is the after occasion. That's after the department. This is what, like, the award ceremony. The owner of Ife came, the founder of OPE came, all the managing director of different banks came. It was a grand occasion, you grab. So it was amazing. It was an important stuff that day. Meanwhile, I have a laboratory work that I should have done, no, but they compelled everybody to come here because it was as a result of what they did in our lab. The individual that funded that particular project in the lab, so the school decided to give him an award. So our department was like, every PG student must be in this venue, so we didn't have any other option. Meanwhile, our rabbit, we've not killed it, so we still have our rabbit in the lab. So what we did was, immediately after, we brought leaves. This is the rabbit feeding. Can you see how the rabbit is chewing? It was amazing. It was very interesting. So we fed the rabbit, we gave the rabbit water throughout that day, because we didn't kill the rabbit that day. We couldn't, because we didn't have the time. So we came the next day, and then we killed the rabbit. We took the blood for experiment. I told you that in this particular, if you've been following my post on this particular channel, I told you that this particular experiment is all about blood fixation. So we started with fixing the blood with glutaraldehyde, and then we fixed the blood also with trypsinization. So we divided the blood into two, where we fix with glutaraldehyde and then fix with trypsin continuously. At some point, as what this guy is doing now, Kenny, is actually trying to wash the blood. You understand? When he's done washing, he will impute it to the centrifuge. We wash this particular blood for 5 different times, and then we centrifuge. When we are done, we try as much as possible to impute it in a conical flask. Are we there now? And then store for the next day, where we try as much as possible to carry out our hemagglutination assay. And it's in this hemagglutination assay that we got out the glutaraldehyde fixed blood. When we're talking about a blood that is fixed, remember, the blood I'm talking about here is rabbit blood. So we're looking at, a semi-solid blood. It's not solid, it's not liquid, So we had it in that particular concentration, and then, we try as much as possible to, use it in hemagglutination assay. So what we did was, already we now have a glutaraldehyde fixed blood and then a trypsinized fixed blood. So we try as much as possible to get a microtiter plate. As you can see, what I'm doing here now, I'm trying to carry out a serial dilution. In this particular microtiter plate, I got out my, glutaraldehyde fixed blood. I took, 100 microliter and impute in one of these particular pores. There are about 24 pores, A to B. So I use A to B for glutaraldehyde, and then I use, C to D for trypsin. So for each particular well in that microtiter plate, it contains glutaraldehyde, buffer, that's PBS, and then my protein sample. And the other, well also contain trypsin, my buffer, and my protein sample. So after incubating for 3 hours, this is my result. That's my laboratory work this week. Thank you for watching.